Very good morning to all of you. A very good morning to all of you. Today I am going to discuss about the sales cycle. Sales cycle was proposed by scientist Howard and Pell. Sales cycle, it is a cyclic representation of event of cell division. What are the different stage of cell division? You know, G1 phase, S phase, G2 phase, and M phase. M phase means mitotic phase. Okay, so it is a cyclic representation of the cell cycle events. That is, they are divided into G1 phase, that is gap 1 phase, then S phase, then G2 phase, and then M phase, that is the actual division phase, that is mitosis or meiosis. Okay, so try to know G1 phase, that is the preparatory phase. G1 phase, S phase, and G2 phase, that include in the interphase. So generally, question come in mind that the cell is whether active in interphase or in sleeping period. What is the answer? Answer is that cell is most actively dividing, means they are most actively prepare the DNA, RNA, and protein to make a sufficient amount before entering into the M phase, that is division phase, that is either mitosis or meiosis. So before entering into the mitosis phase or meiotic phase, the cell is going to prepare the raw materials. What are the raw materials? It can be a proteins, it can be a RNA, it can be a DNA. Means what are the material required to make a cell? So that is G1 means gap 1 phase, and then cell will enter into S phase, that is the most actively active phase where there is a replication of DNA occurs, means synthesis of DNA occurs, and then it will enter to G2 phase. So there is some checkpoints whether the cell have prepared that materials or not. If they have not prepared that material, then it cannot enter into mitosis phase or division phase. So this cyclic representation, that is a cell cycle, it is the G1 phase, S phase, and G2 phase, that is interphase. You can say it is interphase. So most of the time cell prepare the DNA through DNA replication in S phase, and protein and RNA in G1 phase and G2. Okay, so cell division occur in recurrent manner with freedom to fight only at one point in G1. Cell cycle is a progressive event with chance of reproduction. They just synthesis occur. Means you can say that is means making of new daughter cell when the cell is entering into mitosis. Okay, so just I'm briefing that part, that is cell cycle. Okay, so the you see S phase plus G2 phase, that is heterokaryon means synthesis, so DNA repair but no RNA synthesis. So you have to know in which phase of the cell cycle there is a formation of DNA or replication of DNA occur. So that is the S phase. Means in S phase, if there is any breakage in the DNA or there is less amount of DNA, then synthesis of DNA through DNA replication. And if there is a breakage in the DNA, then DNA repair also occur. But no DNA replication occur in G1 phase or G2 phase. So generally G1 phase and G2 phase, there is synthesis of protein. Okay, so next you try to know this phase means cyclic events. First is interphase. So generally the question come in my our mind that interphase, that is the sleeping phase. No, it is not a sleeping phase. It is very active phase. Why it is a very active phase? Because there is synthesis of DNA, there is synthesis of protein, there is synthesis of RNA occur during that phase. So that interphase include G1 phase followed by S phase and then G2 phase. After G2 phase, there is checkpoints. Means the cell will there is some regulatory mechanism through which it is verified that the cell have sufficient amount of DNA, RNA and protein. Then only it will enter into M phase. That is the M phase. Uh, uh, this diagram, you see the starting point, that is the major portion, that is G1 phase. That is, it indicates that it takes maximum duration out of 24 hour. It is minimum 13 and 14 hours. Then it will take 6 to 8 hours like that, and then 3 to 4 hours like that. So generally cell division occur in very short period of time, but preparatory phase is very long. Okay, now M phase means mitosis or meiosis. Not only mitosis, not only meiosis, means division phase. So there are two events I have already told you: the karyokinesis, then followed by cytokinesis. What are the different stages of karyokinesis? Means division of nucleus, means distribution of uh, the uh, chromatids, that is the that contain the DNA. That is the prophase. This one is the prophase, then metaphase, then anaphase, then telophase. These are the similar events either in mitosis or in meiosis. But if you observe the prophase in meiosis, there are two. One is prophase 1 and second is prophase 2. So prophase 2 is just like mitosis. I have already told you that prophase 2 is just like mitosis. But prophase 1 is actual meiosis, where there is a reduction of chromosome. Means chromosome number reduced to half during meiosis 1. So in case of mitosis, only one substage, that is prophase 1, prophase, metaphase, anaphase, telophase. But in case of meiosis, there are two. One is prophase 1, means meiosis 1 and meiosis 2. In meiosis 1, it is prophase 1, metaphase 1, anaphase 1, telophase 1, like that. But in case, if you go through the meiosis 2, that is prophase 2, metaphase 2, anaphase 2 and telophase. But in case of meiosis 1, there is a further subdivision of prophase 1. That situation not come in case of mitosis or meiosis 2. So it is the longest stage in case of meiosis 1, that is leptotin, jagotin, pacatin, diplotin, dikinase. I have already told you these things. It is audible or not? Yes, Yashasvi,
Yes,
Atharva,
sir.
are you listening my sound? So
Yes, sir.
yes, so every cell is going to divide in your body or in the plant body, means during certain process. Okay, so just see the duration of hour in this pie chart. Okay, so that is you the starting point, that is known as G1 phase. G1 phase generally it will take 6 to 12 hours. Means chromosome number is equal to 2n, that is the just like somatic cell. Okay, but if it will enter into the S phase, then it will take 6 to 8 hours. So generally in case of G1, if there is a deficiency of protein or RNA, such type of synthesis occur in case of G1. Okay, so this pies are divided into 24 hours, like that. But if you are dividing bifurcating that different stage, then G1 phase take about 6 to 12 hours. And then after that, the cell enter into S phase, that is most active phase. in which there is a replication of DNA, means synthesis of sufficient amount of DNA occur, because that DNA is going to make a chromosome, chromatid in the for the new daughter cell. So if you see the S phase, that is the very specific phase where it will take 6 to 8 hours and synthesize the DNA by DNA replication. Next come to G2 phase. Whatever the substance left, either DNA or protein, sufficient, means there should be sufficient amount before entering into the M phase. That is M phase, that is the actively dividing stage where mitosis or meiosis occur. If it is reproductive cell, then meiosis occur. If it is somatic cell, then mitosis occur. So whatever the deficiency is there, so there are two checkpoints in G1 and S, and then there are checkpoints just after G2. So what is a checkpoint means whether there is a deficiency of DNA or deficiency of RNA or deficiency of protein are there in the cell. If there is a deficiency, then cell will not enter in M phase, that is actively divided stage. I'm talking. Okay. So try to know what is cell cycle. That is the cyclic events of the G1 phase, S phase, G2 phase, and mitotic phase. In mitotic phase, you are observing the prophase, metaphase, anaphase, telophase. But if you are observing the meiosis, then there is the further two division, meiosis 1 and meiosis 1. Just after completion of meiosis 1, it will enter into cytokinesis and then come to meiosis 2. Then there will be completion of meiosis. Otherwise, there is no completion of. But you are observing, that is the arrow. Are you observing G0 phase, resting phase? If cells do not want to synthesize any raw materials like DNA, RNA, or protein, or if they do not want to divide, means they do not want to enter in mitotic phase or meiotic phase, then it will come to G0 phase, that is resting phase. Okay. So that is the quiescent, means dormant phase, where there is no preparation of any raw materials for the further mitosis, meiosis, or it is not going to enter into division phase, that is G0 phase, that is resting phase. G1 phase, that is growth and metabolism occur. Means metabolism means synthesis. You know what do you mean by metabolism? Synthesis means synthesis of RNA, synthesis of protein, that is occur in G1 phase and G2 phase. But if you are observing the S phase, on maximum amount of RNA DNA replication occur. What do you mean DNA replication? Making of DNA from same DNA sample. Means old DNA, from old DNA we are going to prepare the new DNA in presence of DNA polymerase. Enzyme is DNA polymerase. Then how the synthesis of RNA occur in presence of RNA polymerase and protein. You know protein, I will discuss each and every topic in detail in your coming chapter. Okay. So just try to learn what do you mean by G0 phase, that is the resting phase where there is no preparation of material or no division. The cell will not enter into cell division, or it is not going to prepare any raw material like DNA or protein or RNA. So generally it is resting phase. But if you are observing the G1 phase, that is growth and metabolism. What do you mean metabolism? I have already told you that it is actively division, means synthesis phase, where there is synthesis of either RNA or protein or DNA. So that is generally in case of G1, there is synthesis of protein as well as synthesis of RNA. So that's why it is known as growth and metabolism. And as that is DNA replication, means whatever the DNA means you are going to, the cell is going to prepare two cell from one cell. So automatically there is sufficient amount of DNA requirement, that is for making new cell, because each cell require the DNA and protein and further RNA. So that is required. So that should be sufficient amount in new baby cell, that is daughter cell. So that's why the cell will synthesize the sufficient amount of DNA in S phase. And you must know the duration. The maximum duration is of G1 phase, that is 6 to 12 hours. If you are going to divide the 24-hour, then 6 to 12 hours generally cell are in G1 phase, that is growth and metabolic stage, and 6 to 8 hours the cell are in S phase for making the DNA, and then 3 to 4 hours cell is going for making the whatever the substance deficient in a small quantity they are synthesized. Then cell will enter into M phase for actively division. Means M phase include prophase, metaphase, anaphase, telophase. So this is the pie chart representation of cell cycle. Next, I am talking just this is the representation means the length of time duration, that is G1 phase has maximum duration, that is 6 to 12 hours, then S phase has 6 to 8 hours, then G2 phase at 3 to 4 hour, then M phase that is maximum 1 hour, and in further there is subdivision of the time. You see that is the length of time for the division. So that is prophase have maximum time, it will take maximum time, then telophase, then metaphase, and then anaphase. So the but sequence is that, means it is a it is depicting the how much time each stage is taken, means in descending order, the duration of the time taken by the cell in each stage, that is in descending order. Okay, so maximum duration taken by G1 phase, then S phase, then G2 phase, then M phase. In M phase also there is further subdivision for the time duration, that is maximum time taken by prophase, then telophase, then metaphase, then least time taken by anaphase. Okay, so but the sequence for the mitotic division is prophase, metaphase, anaphase, telophase. This is only for time taken by cell in each cell. Okay, but the sequence of the event is first prophase occur, then metaphase occur, then anaphase occur, and then telophase occur. So just you must remember these things, the duration of time in descending order, which stage is taking a maximum duration of time. Okay, so it is clear to all of you, ये दिख रहा है आपको? Now come to I phase, interphase. I phase means interphase, and M phase means mitotic phase. So what is the difference in I phase and M phase? Try to know what is the difference between I phase and M phase. So are you listening my sound? Yes, Ashish,
आपकी आवाज आ रही है
आ रही है आवाज?
सर सर
आवाज आ रही है कि नहीं बताओ?
सर
कट कट के आ रही है?
सर क्लियर आ रही है, आवाज क्लियर आ रही है।
वेरी गुड। तो आपको समझना है कि व्हाट इज डिफरेंस बिटवीन आई फेज एंड एम फेज। आई मींस आई बोले तो इंटरफेज, एम बोले तो एम पहले माइटोटिक फेज और म्यूटिक फेज। एम से माइटोसिस भी हो सकता है, एम से म्यूसिस भी हो सकता है। तो मेटाबॉलिकली रेस्टिंग फेज, दैट इज एम से है मेटाबॉलिकली रेस्टिंग फेज, but cytologically most active. M means that is the mitotic or meiosis and metabolically resting. Okay, so you say the interphase means it is not a resting phase. What? Why? Why? Why it is not a resting phase? It is the most metabolically active phase. Why? Because in I phase means interphase there is synthesis of DNA, there is synthesis of protein, there is synthesis of RNA. That's why you can say it is a most active, metabolically active phase. But in term of cytologically um, uh, activity, it is less cytologically activity occurs, means division of the cell occurs. So no division of cell occurs, that's why it is known as cytologically resting. So this is the difference. Which one is the most active phase? That is I phase, that is interphase. Why? Because that is the phase in which the cell is going to prepare all the raw. So just think about a pregnant lady. They are taking sufficient amount of food just to prepare their baby. They are taking a sufficient amount of calcium for making the bone formation of the baby child. Okay, and all the vitamins, all the minerals, and sufficient amount of protein just to prepare the baby. Okay, in the same way in interphase also, it is the preparatory phase where there is synthesis of protein occur, where there is synthesis of RNA occur, where there is synthesis of DNA occur. So that include G1 phase, F phase, and G2. So if in the sense of preparation of material, that is the most actively phase of the cell cycle, that is I phase. And in M phase, there is no synthesis of any material occur. Whatever the DNA or RNA or protein synthesized in I phase, that is going to utilize and make two daughters by karyokinesis followed by cytokinesis. So karyokinesis is cytologically most active phase, but G1 is metabolically and biochemically most active phase. Why? In G1 phase, there is synthesis of maximum amount of protein and yes, and RNA. But if you are observing the N phase, that include all prophase, metaphase, anaphase, telophase, then the prophase, that is the means there is a division of nucleus occur. Further, there is a division of cytoplasm. So first event, that is the karyokinesis and then cytokinesis. Karyokinesis take maximum duration during the phase, and cytokinesis process that take least time for division of cytoplasm. So try to know what is difference between I phase means interphase and M phase means metabolically active resting phase, that is mitotic phase, or meiotic phase, that is dividing phase, division phase. So actual division phase is M phase, but if you say I phase, that is metabolically active phase where there is no division of cell. So this is it is clear to all of you. It is clear to all of you or not? Yes. Now come
Correct.
to yes. There any question? Any
सर
question?
एक बार
Any?
दोबारा समझा दो सर, हल्का हल्का।
मैं ये बताना चाह रहा हूँ कि जस्सी मैं आपको मैंने पहले बताया है। देखिए यहाँ पे ये जो है, ये जो दिख रहा है आपको फ्लो चार्ट, ये डायग्राम दिख रहा है, साइक्लिक डायग्राम। तो
यस सर।
यस, तो हम कौन कौन से स्टेज को हम लोग बोलेंगे कि ये इंटरफेज है। तो इंटरफेज दैट इज द G1 फेज। ओके, interface that is the G1 phase, then S phase and G2 phase. So there is no cell division occur in during that period. There is no cell division occur. What is going on means preparation of any materials, means just like a pregnant ladies, inside that there is a formation of new baby. So what that requires sufficient amount of food that is rich of vitamins, minerals, calcium, magnesium, all the vitamins, all the minerals and iron. So that why? Because they are going to she is going to synthesize new baby, is giving life. In the same way, the cell is going to prepare all the raw material just to make a two daughters. Are you listening, my son? Means the cell is going to prepare all the raw materials. What is the raw materials? The proteins, that if there is no protein, you cannot expect the formation of two daughters. If there is no sufficient amount of DNA, you cannot expect that there is a formation of new baby cell, daughter cell. And if there is no RNA, so that synthesis generally occur during G1 phase, S phase, and G2 phase, that constitute together known as interphase. So interphase means it is not a sleeping phase, it is a very active phase, that is G1 phase, S phase, and G2 phase. So generally in our mind it is saying that I phase, that is interphase, is very inactive phase. We cannot say it is not an inactive phase. It is a very active phase for making a new DNA molecules for a new daughter cell. After preparation of all these materials, cell is going to check by some system, regulatory system, CDK1 and CDK2. Okay, so whether that sufficient amount of material is there inside the cell or not. If there is deficiency, then in the G2 phase, all the material will going to synthesize, so that it should be in sufficient amount. After getting the sufficient amount of protein, DNA, and RNA, then only cell will enter into M phase. So M phase, the first event of the M phase is prophase, then metaphase, then anaphase, then telophase. So that include the karyokinesis followed by cytokinesis. Then again cell, daughter cell will feel there is a deficiency. What is the deficiency? Deficiency of maybe some amount of RNA, some amount of DNA, or some amount of protein. So this cell again it will enter into again in G1 phase to make the sufficient amount of protein, sufficient amount of RNA, and then again enter into S phase for getting multiply sufficient amount of DNA. So that process is going on every time. Okay, and then only we are getting the new cell. Otherwise we cannot expect the new cell, because raw material is required. Means if you have a raw material, only then we can expect the product. Otherwise you cannot expect the product. If you have no raw material, then how can you? How it is feasible to make a daughter cell? No, cell cannot produce two daughter cell unless and until there is sufficient amount of DNA, RNA, and protein. It is clear? ये दिख रहा है आपको? इसी तरह से ये जो है आपको जो मैं बता रहा हूँ, ये भी वही वाला स्टेज दिखा रहा है। ये कह रहा है कि ये सेल तब जाएगा एम फेज में जब इसके पास सफिशिएंट अमाउंट में डीएनए, आरएनए, प्रोटीन होगा। तो कितना टाइम लगाता है, ये कितना टाइम लगाता है? G1 फेज कितना लगाता है, एस फेज कितना लगाता है, G2 फेज कितना लगाता है, तब जो है ये इंटर करता है एम फेज में। ये तब इंटर करेगा जब इसके पास सफिशिएंट अमाउंट। लेकिन जब सेल को जरूरत नहीं है, न डीएनए बनाने के, न आरएनए, न प्रोटीन, और न ही सेल डिवीजन का, तब वो किस स्टेज में चला जाता है? G0 स्टेज, मतलब रेस्टिंग स्टेज। ये न तो डीएनए, आरएनए, प्रोटीन बनाता है और न ही सेल डिवीजन करता है, तो उसको कहते हैं G0 स्टेज। इट इज क्लियर? ये क्लियर हुआ कि नहीं, ये
यस
बताओ।
सर। यस
हाँ,
सर।
तो ये सीक्वेंस जो बता रहा है, ये सीक्वेंस बता रहा है क्या? फर्स्ट इवेंट G1, सेकंड इवेंट, ड्यूरेशन ऑफ टाइम, कि सबसे ज्यादा कौन टाइम लेता है? G1 लेता है। उसके बाद कौन सा लेता है? S लेता है। उसके बाद कौन सा लेता है? G2 लेता है। इन आवर, आई एम टॉकिंग इन आवर। और ये एम फेज जनरली 1 आवर लेता है, और 1 घंटे में इसका भी फर्दर मिनट में डिवीजन है। प्रोफेज सबसे ज्यादा लेता है, उसके बाद टेलोफेज लेता है, इसके बाद मेटाफेज लेता है, इसके बाद एनाफेज लेता है। लेकिन ये नहीं बता रहा ये सीक्वेंशियल इवेंट है, ये टाइम बता रहा है। सीक्वेंशियल इवेंट क्या है कि G1 S G2 M। अब M में प्रो मेटा एना टेलो, प्रो मेटा एना टेलो, समझ में? पहले इवेंट्स क्या है? प्रोफेज होगा, उसके बाद क्या होगा? मेटाफेज होगा, उसके बाद क्या होगा? एनाफेज होगा, उसके बाद होगा टेलोफेज। ये बता रहा है सीक्वेंस की कौन कितना टाइम लेता है। सबसे ज्यादा प्रोफेज लेता है, उससे कम लेता है टेलोफेज, उससे कम लेता है मेटाफेज, उससे कम लेता है एनाफेज। ये क्लियर हुआ कि नहीं इतना? इट इज क्लियर अप टू दैट?
Yes, sir. Yes,
यस।
sir.
अब मैं ये कह रहा हूँ कि G0 फेज जो मैं बता रहा था, इट इज कॉल्ड फेज ऑफ सेल डिफरेंशिएशन। इन दिस फेज सेल क्वाइट फ्रॉम द सेल साइकिल से वो बाहर आ जाता है। ऑल्सो कॉल्ड फेज ऑफ सेल क्विसेंस, मतलब कि ये डोरमेंसी में चला जाता है, ना वो मटेरियल सिंथेसाइज करता है और ना वो सेल डिवीजन में जाता है, दैट इज नोन एज क्विसेंट। ओके, तो क्विसेंस ऑफ द सेल कैन बी रिवर्सेबल और इरिवर्सेबल, या तो वो एक बार चला गया तो फिर से रिटर्न बैक नहीं होगा, उसको कहेंगे इरिवर्सेबल, और फिर से वो आ जाएगा जस्ट फॉर इंटरफेज और फॉर माइटोटिक, ठीक है ना। तो रिवर्सेबल क्वेश्चंस इज फाउंड इन प्लांट्स, रिवर्सेबल किस में पाया जाता है? प्लांट में। वेयर एज डिफरेंशिएटेड सेल कैन शो डिफरेंशिएशन एंड इंटर इन द सेल साइकिल। इरिवर्सेबल क्वेश्चन इज रिपोर्टेड इन एनिमल, मतलब कि ये जनरली एनिमल में दिखाता है इरिवर्सेबल क्वेश्चंस। क्वेश्चंस मतलब क्या होता है? सपोज कि सीड है, सीड को हम इस ईयर में आपने ग्रो करा के मैंने पौधे में से सीड निकाला, अब उसको हम रखते हैं, तो ये सीड जो है डोरमेंट पीरियड में जस्ट फॉर नेक्स्ट सीजन यू आर गोइंग फॉर सोइंग, then you get the plant. It is the same situation like dormancy of the seed. Okay, so generally plant cell reversible क्वेश्चंस दिखाता है और एनिमल सेल इरिवर्सेबल क्वेश्चंस दिखाता है, मतलब कि G0 मतलब क्वोशेंट फेज, दैट इज द डोरमेंट फेज where there is no activity occur, either synthesis of raw material like DNA, RNA, protein or division of the cell, मतलब दोनों इवेंट्स जो है एब्सेंट रहेगा, किसमें? जब सेल G0 फेज में चला जाएगा। अब देखिए यहाँ पे G1 फेज क्या है? फर्स्ट ग्रोथ फेज। ये फर्स्ट ग्रोथ फेज है जो हम बता रहे हैं, इंटरफेज का जो फर्स्ट पार्ट है। मैं बताया ना इंटरफेज में कौन कौन होता है? पहला इवेंट है G1, दूसरा इवेंट है S, तीसरा इवेंट है G2। तो मैं ये बताना चाह रहा हूँ कि क्या क्या होता है G1 फेज में, बनता क्या क्या है? डी एंड टीपी, मतलब कि बिल्डिंग ब्लॉक। जैसे दीवार बनता है तो दीवार के लिए ब्रिक चाहिए ना, वॉल के लिए। इन द सेम वे डीएनए बनता है तो डीएनए के लिए रॉ मटेरियल चाहिए, ये चाहिए डी डी एटीपी, डीजीटीपी, डीसीटीपी या डीटीपी। इसके बाद अमाइनो एसिड का सिंथेसिस होता है, उसका सिंथेसिस साइक्लिन प्रोटीन होता है, एक्टिव जीन एक्सप्रेशन होता है और एनर्जी करेंसी बनता है। तो ये आपको ध्यान रखना है क्या क्या बनता है। प्रोटीन बनता है, अमाइनो एसिड बनता है, उसके बाद जीन का एक्सप्रेशन होता है, आरएनए सिंथेसिस हम बार बार बता रहे हैं। फिर जो जस्ट लाइक ब्रिक फॉर मेकिंग वॉल, in the same situation here for the DNA that is required, that is a unit, that is the ATP, GTP, CTP, TTP, AGCT. Okay, so that is synthesized in G1. Okay, it is clear. Now come to S phase. S phase is very active phase where there is synthesis of DNA. Means you are getting the identical copy of DNA from same copy. मीन्स यहाँ क्या क्या होता है? डीएनए इज कॉपीड और रेप्लिकेशन ऑफ डीएनए अकर। और दूसरा है हिस्टोन प्रोटीन सिंथेसिस अकर, दूसरा हिस्टोन प्रोटीन का सिंथेसिस होता है। तीसरा सेंट्रियोल सिंथेसिस अकर, मीन्स फॉर्मेशन ऑफ सेंट्रोमेयर, सेंट्रोमेयर होता है। सेंट्रियोल सिंथेसिस अगर फॉर द फॉर द क्रोमोजोम, सेंट्रोमेयर, सेंट्रोमेयर सेंटर में होता है, काइनेटोकोर। ओके, क्रोमोजोम में जो मैं बार बार बताते रहता हूँ, सेंट्रोमेयर या काइनेटोकोर, बोथ आर। ओके, तो ट्राई टू नो क्या क्या सिंथेसिस होता है इस पेज में। डीएनए सिंथेसिस होता है, दूसरा हिस्टोन एक टाइप ऑफ प्रोटीन है जो कि डीएनए के साथ रहता है हमेशा, तो ये भी बनता है। तीसरा सेंट्रियोल सिंथेसिस, दैट इज यूज इन मेकिंग द क्रोमोजोम, तो दैट इज द रिक्वायरमेंट फॉर, दैट इज द ड्यूरिंग सिंथेसाइज ड्यूरिंग एस फेज। तो G1 फेज के बाद कौन सा फेज आएगा? एस फेज। एस फेज के बाद कौन सा फेज आएगा? एनीवन? G2 फेज। ओके, एस फेज के बाद आएगा G2 फेज। So just see what event is going on, that is S phase में क्या होता है? Synthesis occur, the DNA synthesis occur inside the nucleus. Okay, so just try to see that animated diagram and slide. That is next stage is G2 phase. अगर यहाँ पे कुछ भी बच गया बनाना, तो यहाँ जो कुछ भी डिफिशियंसी हो जाएगा उसकी सिंथेसिस होगा, मतलब अगर ब्रेकेज हो गया डीएनए में तो वहां रिपेयर हो जाएगा, with histone DNA blockage to form the chromatin। मतलब ये क्रोमेटिन जो है ये बना होता है हिस्टोन प्रोटीन से और डीएनए। इतना समझ लो कि क्रोमोसोम का जो दो कंपोनेंट है कौन कौन सा है, हिस्टोन
सर
एंड डीएनए। हिस्टोन प्रोटीन है और डीएनए यू नो न्यूक्लिक एसिड and repression of gene activity transcription began completed up to enough means uh, uh, gene activity means transcription means whatever the DNA or RNA synthesize that is going to rearrange मतलब DNA और प्रोटीन आपस में rearrange होगा for what, for making chromatin, that chromatin that form the chromosome। तो ये आपको समझना है कौन कौन से इवेंट्स में कौन कौन सा बायोमोलिक्यूल का सिंथेसिस होता है। जनरली जीवन फेज में प्रोटीन बनता है, देन अमाइनो एसिड बनता है, देन डीएनटीपी बनता है, डीएटीपी, डीजीटीपी, डीसीटीपी, डीटीटीपी। क्यों? क्योंकि डीएनए बनना है एस फेज में। एस फेज में जब बन हो गया कंप्लीट डीएनए और हिस्टोन प्रोटीन, तब वो जी2 फेज में जाता है। जी2 फेज में जो भी रिपेयर होना है डीएनए का रिपेयर होगा, हिस्टोन और डीएनए अरेंज होके क्रोमेटिन बनेगा जिससे क्रोमोसोम बनेगा। अब रिप्रेशन ऑफ जीन एक्टिविटी होगा, बिगिंस कंप्लीटेड अप टू एनाफेज, मतलब कि अब ना डीएनए बनना है ना आरएनए बनना है। क्यों? क्योंकि अब डिवीजन फेज में जा रहा है सेल, ठीक है ना। ये दिख रहा है आपको, everyone are observing that sketch of cell cycle। पहला क्या है, इंटरफेस। ये कहां से कहां तक है, जीवन से जी2 तक। तो जीवन में दैट इज सेल इज गोइंग टू मैच्योर, or means there is a protein synthesis, there is a amino acid synthesis, there is a small unit like DATP, DGP, DGTP, DTTP, these are the synthesized, and then all the requirements is going to fulfill in G1 phase, that is metabolically active phase। Then making of DNA through process of replication occur, then synthesis of histone protein occur in S phase। अब यहाँ पे क्या हो रहा है, whatever the DNA, there may be some damage in the DNA that is going to repair। अब जो है यहाँ पे भी ब्रेक लगेगा, किसका, ना डीएनए बनेगा ना आरएनए बनेगा, तो जीन इज गोइंग टू रिप्रेस बाय दैट सम एक्टिविटी। सो इन केस ऑफ जीटी, क्यों, क्योंकि अब जो है कुछ भी बनना नहीं है, क्या, ना डीएनए बनना है ना आरएनए ना, तब जो है एक्टिवली डिवीजन स्टेज में जाएगा, प्रोफेज, प्रोमेटाफेज, मेटाफेज, एनाफेज, टेलोफेज, फॉलोड बाय साइटोकाइनेसिस। जैसे ही ये कंप्लीट हो गया सेल डिवीजन, then it will enter into G1 phase again। ओके, और अगर उसको न आरएनए बनाना है, ना प्रोटीन बनाना है, ना डीएनए बनाना है, ना ही सेल डिवीजन में जाना है, तो G0 फेज में आ जाएगा। ये आपको मैंने बताया आज। नेक्स्ट डे आई विल टॉक अबाउट चेक पॉइंट, कि व्हाट आर द चेक पॉइंट, CDK1, CDK2, दैट इज द टू चेक पॉइंट that check whether the all the material, raw material is synthesized by the cell or not। If it is synthesized, then allow for next phase, otherwise it will, that cell have to synthesize that material unless and until that is become sufficient, then it will enter into the division phase, that is mitotic phase, that is the division phase, that is either mitosis or meiosis। So this is the very well diagram, well sketch of the cell cycle। Okay, so have you any question, any question, कोई क्वेश्चन है तो बताओ।
नो सर।
थैंक यू, and I will explain the checkpoints in the next class। Okay।